I would like to welcome to our series of presentations on the Global Food Systems Seed Grant applications. We have here Dr. Aakeroy, who is a professor of chemistry in the College of Arts and Sciences here at Kansas State University. His work was carried out with Dr. Ganga Hettiarachchi, professor in agronomy in the College of Agriculture. The topic of today's presentation will be exploring co-crystal technologies for efficient and sustainable nutrient management. Dr. Aakeroy?
Thank you, Maureen, for that introduction. Yes, I'm here today to tell you a little bit about the project that was made possible by a seed grant from the GFS, and this is a collaboration between Department of Agronomy and Department of Chemistry, and you can see the names of all the participants on the screen up at the very front here. So as everyone is fully aware of, the presence of fertilizers, or the need for fertilizers, is ongoing and increasing with the fact that the population of this planet is growing steadily. And the purpose of this particular project was to try to figure out if there is any way that we can better manage nutrients, in particular nitrogen, as part of the cycle where we put fertilizers on the crops, and then how best, or how we can facilitate the way in which the plants take up those fertilizers. First things first, the molecule that we are interested in, and I'm a chemist, so we're going to be talking about molecules. So it's a simple molecule for those of you who are watching, and urea is the molecule that we're interested in. It's a small molecule, as I said. It's interesting because it has a lot of nitrogen per molecular weight, which is kind of important. So nitrogen is one of the key components in any fertilizer today. And I just pulled out a couple of sort of overview slides. This slide is a little bit, or the data here is a little bit old, but this will give you some sense of how much fertilizer we use globally. Now, this is about 8 years old, and you can see that we are focusing on 3 different types of contributors to fertilizers. Nitrogen is by far and away the biggest. Then we also have phosphate and potassium. And I think the latest numbers for nitrogen is that we are using approximately 200 million tons of nitrogen fertilizer every year on this planet. Now, out of that 200 million tons, almost 60% comes in the form of urea. So the little molecule that you see in the top right corner is by far and away the most common nitrogen delivery in the fertilizer industry. The problem with urea, though, is, well, there are some pros and cons. So there are good parts of urea. So first of all, it is relatively inexpensive, and it has a high nitrogen content, as I pointed out. So per molecular weight, there's a lot of nitrogen. So nitrogen is indicated by blue in this particular picture. But there are some cons as well. And unfortunately, since we often tend to over-fertilize because the plants cannot readily absorb the nitrogen that we're putting in, the use of fertilizers tend to produce a large amount of greenhouse gases. There is eutrophication that is taking place, which means that we have problems with environmentally detrimental impacts on freshwater, lakes, rivers, and even oceans as well. Part of the problems with urea come from the fact that it's almost too soluble in water, which means that it's going to break down too quickly, and plants do not have time to absorb the nitrogen that we want to deliver in an efficient way. The second point of urea is that it's also hygroscopic. Now, if you look at the top left-hand corner, you see a really nice white crystalline powder. But if you leave urea out in humidity, it becomes a sticky liquid, which means that storage and transportation becomes relatively difficult. Now, on top of that, and this is a very recent development, the price of fertilizers, broadly speaking, has gone up dramatically, and I'm going to talk a little bit more about that later on. The recent developments in the Ukraine and the Russian border has essentially taken the price of urea up about $800 per short ton. It makes a huge difference. Still inexpensive, relatively speaking, but given the fact that we're using about 200 million tons a year globally, you can quickly calculate that there's a lot of money spent on fertilizers, and we would like to be able to do that as efficiently as possible. So what happens then if you put urea onto the crops in the field? So this is quite a busy slide that I just want to draw your attention to a couple of things. Ideally, we want all of the nitrogen in the urea molecule to eventually end up in the plant. But that unfortunately doesn't happen. So as soon as urea is put into the soil, if there's any moisture present, naturally occurring enzymes will begin to break down urea into a wide variety of nitrogen-containing compounds, starting with ammonia, which is NH3 in this slide. And NH3 is a gas, so that's going to be essentially escaping from the soil, and the plant will never have a chance of absorbing that gas at all. And then there is a second step, and that's the nitrification. And these kind of breakdown processes are carried out by naturally occurring enzymes. So if we want to try to maximize plant uptake, ideally we would like to, first of all, slow down the hydrolysis, meaning the breakdown of urea. And secondly, we want to try to limit the nitrification process, because we do not want to produce nitrates, NO3-, because that will lead to unwanted fertilization of fresh water, lakes, rivers, etc. And it will also produce a large number of greenhouse gases like nitrous oxides, NO2, and other gases as well that are harmful. So ideally, we want to try to optimize the transformation of urea to something that the plants can take up. And that's relatively complicated. The take-home message from this slide really is that even though we're putting a lot of urea onto the crops globally, only a relatively small fraction of the nitrogen in those urea molecules end up in the plant itself. And we would like to try to optimize that process, because if we can, then we can reduce the amount of fertilizer that we put on the fields in the first place. So the point of this, or the premise of this project, is that if we can decrease the solubility of urea in water, we will slow down the release, and then we can make a more efficient transformation of urea into nitrogen that can be taken up by the plants. So the two-step process that we're aiming for is to go from urea on the far left here. We want to slow down that process as much as possible, so we can have a slow delivery over an extended period of time, and that will limit the amount of ammonia gas that we produce. And that ammonia gas the plants can't use at all in the first place. And then we can produce more ammonia, NH4+, which is in a form that can be absorbed by the plants. Now, it has been established previously that urease, which is the enzyme responsible for breaking down urea, is going to be limited or hindered by the presence of acids or organic acids in the soil. So a lower pH is going to reduce the enzyme activity. So we're going to have to keep that in mind at some point. Secondly, we also want to try to stop the nitrification process. Once we've gone from ammonia to ammonium, we want to see if we can accumulate the NH4+, and we want to stop the nitrification process. And again, a variety of acids, organic acids, have been shown to slow down the accumulation of nitrates. So there are two things that we would like to accomplish here. First of all, that we would like to try to decrease the solubility of urea in water, and we would like to increase the stability of urea so it doesn't go from a crystalline solid into a sticky liquid for storage purposes, life or shelf life, and things of that nature. So the way we're going to solve this problem is relatively simple. So we are using something called the co-crystal technology, which we have developed for probably about 15 years in my lab now, but we have never really applied it to agrochemistry or agriculture before. So the basic principle is that we're going to take urea, which has high solubility and it's highly hygroscopic. Both are disadvantages in this process. And we're simply going to combine urea with a coformer. And the coformer in itself is going to have low solubility in water. It is not going to be hygroscopic, it's going to be cheap. It needs to be readily available. Obviously, it needs to be non-toxic, no harmful side effects. And ideally, if we can add some nutritional benefits to the plant within the coformer, that would obviously be additional benefit. So we're going to synthesize these two. It looks simple, it's just A plus B, but it turns out that it's not quite that simple. But we have been able to develop guidelines for how we can accomplish that. And the hope is that by combining these two components, we would end up with a new solid, the co-crystal of urea, which has lower solubility in water and which is also more stable with respect to humidity. So a quick overview of the project. There are essentially two parts of this. This is the chemistry part. So in the Department of Chemistry, we're going to try to make new solid forms, new co-crystals of urea. And we're going to focus on the combination of urea with a series of organic co-crystals. You see 6 of them listed in the bottom left corner here. Hopefully, the goal is to try to tune or fine-tune the solubility in water, and also to provide some sort of pH control, because we don't want the pH to be too high. We don't want the soil to be too alkaline, because that's going to promote urease, which will subsequently break down urea. The second part is that we want to try to make some inorganic or ionic co-crystals that will contain not only nitrogen, but also phosphate. So we will have a dual mechanism, or a mechanism for dual delivery of 2 of the key fertilizer ingredients, both nitrogen and phosphorus. So this is essentially part 1, and this has been carried out in the Department of Chemistry. Part 2 is going to be trying to figure out what happens to the nitrogen if we deliver a fertilizer in the form of a urea co-crystal as opposed to of urea in and of itself. So 2 questions that we want to try to address. Will these new solid forms of urea inhibit or slow down the hydrolysis, the step 1 of urea breakdown? And secondly, will those new co-crystals of urea inhibit or slow down or limit the subsequent and highly undesirable nitrification of nitrogen-containing compounds? And this work was done in the Department of Agronomy. So first things first, the synthesis of urea co-crystals is relatively simple. You can actually accomplish this using a mechanochemical principle. So you don't need a lot of beakers and solvents. So in that sense, it's a very green process. So you mechanically mix urea with a co-former, and you do a little bit of solution chemistry. You slowly evaporate the solvent, and you end up with a solid material that's going to be the co-crystal. And we typically analyze the solids using a variety of characterization methods. Single crystal X-ray diffraction will basically provide us with an image of molecular resolution to find out exactly where the different molecules are with respect to each other. And we can find out exactly if urea has bound directly to the co-former. And then we use powder X-ray diffraction to analyze the bulk of the material. And we use some additional methods to investigate purity and extent of reaction and so on and so forth. So this is just one picture on the top right corner. This is a co-crystal that we made in the lab, and this is a combination of urea. And hopefully you remember that small molecule with 2 blue atoms. So we have 2 molecules of urea and 1 molecule of pimelic acid. So it shows that we have a 2-to-1 relationship between, so for every 1 pimelic acid molecule, we have 2 urea molecules. And that's good because we have, relatively speaking, more nitrogen per overall molecular weight. And then just some diagrams or diffraction patterns that shows that the whole bulk material is homogeneous and pure. And then we do something similar for the ionic compounds. This is the mechanochemical synthesis of urea. And you can see on the right-hand side, we see a crystal structure, meaning single crystal X-ray diffraction, which shows us the orientation of different molecules with respect to each other. On the left-hand side, we're looking at powder diffraction patterns of urea by itself, the different reactants by themselves, and then subsequently diffraction patterns of the new product. And again, we can show that the product is pure, it doesn't contain any unwanted peaks, so it's a homogeneous solid. The second part is very, very similar. Bottom left corner, again, a crystal structure that shows the orientation of molecules and ions with respect to each other. So basically provides a 3-dimensional map of exactly what happens between different molecules and ions in this solid. The diagrams on the right-hand side in the bottom right corner will show us information about the overall homogeneity and purity of the bulk material. That's all very well, but once we've made these co-crystals, the question is, did the physical properties change? So keep in mind that we're aiming for 2 things. We are first of all hoping to reduce the aqueous solubility of urea in
the aqueous solubility of urea, because that will hopefully slow down the breakdown process and give the plants more time to absorb nitrogen. And let's see what happens then. So this is, we just have a concentration on the y-axis, and in this diagram we can show that urea has very, very, very high solubility, and the unit here is moles per liter. So it's approximately 17 moles per liter of urea in water. That is really, really soluble. And then we look at the solubility of urea and pimelic acid. As you can see, the bar in that histogram is much reduced. And basically, we have reduced the solubility in water of urea by a factor of 20 times. And if you go to the far right-hand side of that diagram, you can see that we have decreased the solubility of urea 66-fold, which is a substantial increase, which is exactly what we were hoping for, which means that in principle, you should be able to distribute a substantially smaller amount of urea, but by virtue of having it released more slowly over an extended period of time, you might be able to decrease the total amount of urea and fertilizers that you put on the crops over an extended period of time. So that's one thing. The other thing is that we wanted to reduce the sensitivity to humidity, and let's see what happened there. So urea is, and it looks like it's sort of a liquid. So this, if you remember from the very first slides, you had a very, very nice crystalline white powder of urea by itself. Again, upon exposure to humidity, then it turns into a sticky liquid. Whereas on the far right-hand side, you see the simple photographs of urea co-crystals that have been left in the same kind of humid conditions over weeks, if not months, and they are rather insensitive to humidity, which means that storage and transportation and essentially shelf life is going to be much improved. So we have, to that extent, or to that point, been able to rather effectively improve the physical properties of urea using the co-crystal technology. Now, what about the soil incubation studies? Well, this is going to tell us a couple of things. So if you see on the left-hand side of this slide, we are trying to address two particular issues. So first of all, there are two enzymes that are operating naturally in the soil, and those enzymes will hydrolyze urea, which means that the enzyme is going to break urea down. Some of that is going to end up in the plant, but we want to try to control the hydrolysis in order to maximize plant uptake. We also want to try to limit the nitrification, which is also done by another enzyme. And those are the two things that we want to focus on in these soil incubation studies. And all of this work was done in the Department of Agronomy. In addition, there's a clever setup that Hettiarachchi's group has developed, which will allow them to capture any kind of ammonia that is being released in the process. I'm going to quickly just walk you through how this is going to work. So this is the experimental setup. So you get some soil and you pack the soil. So this is going to be like a mimic of soil that you find in a variety of fields, and you pack that into a petri dish. And you can see those petri dishes in the middle of this slide on the far right-hand side. You can basically see that they apply the treatment, whether it's a control experiment or urea itself or the co-crystals thereof, and you apply that in the center of the petri dish. And basically, we're going to look at a spatial movement of the nitrogen throughout that petri dish, which will be more clear on the subsequent slide. Water is added to make sure that it's sufficiently moist, because that is needed for the hydrolysis. And basically, we're looking at, so it's a little bit too fast. So once you have packed the petri dishes with soil, applied the formulation, you seal the petri dishes, secure with paraffin, and then you place those petri dishes in a larger beaker or in a larger dish, which has been coated or equipped with a boric acid soaked sponge. So the boric acid is essentially going to absorb any ammonia that is given off. And after that, they wanted to examine whereabouts within this petri dish that the nitrogen is going to move to. And you can see that basically they will look at separate segments of the petri dish, further distances further and further and further away from the centrally administered fertilizer. So measurements are going to be done. So basically the boric acid is going to capture any kind of ammonia that is being extracted. We're also going to use potassium chloride extraction, which basically will capture ammonium, NH4+, nitrite, which is NO2-, and nitrate, which is NO3-. So using these different methods, as I said, the boric acid will capture the ammonia, and they have a very clever flow injection analyzer system that will effectively capture all the different types of nitrogen-containing compounds that are going to be produced once the urea and the urea co-crystals are exposed to the soil and the enzymes in the soil. So the results will tell us a couple of important things then. So the first data slide I'm showing from the group in agronomy will tell us about the release of volatile ammonia. Again, the idea here is that we want to try to limit the amount of ammonia gas that is being released, because ammonia cannot be effectively utilized by the plant. So in the diagram, you can see that urea, the control, doesn't have any release of ammonia, as expected. Urea releases some of it, and then you see co-crystals, 1, 2, 3, 4, 5, 6 different co-crystals to the right of urea. A couple of the co-crystals on the far right-hand side releases more ammonia, and we are looking into the reasons for that. But 4 of the co-crystals, BUS, NPU, BUP, and BUF, all have reduced the amount of ammonia that is released into the atmosphere, which is a good thing. And you can see that BUP, for example, basically has a limited almost any kind of ammonia volatilization in this particular process. So that is a great step forward. Now, ammonia release is only a small part of the release of these compounds. So if we're looking at other types of, so this is basically the hydrolysis or the nitrification part. So keep in mind, the whole goal of this is to try to reduce the amount of nitrate, or the nitrification part, that we're going to be releasing. And as you can see, the hydrolysis, which is described on the left-hand side, urea is almost to the far left, and the co-crystals do not necessarily slow down the production of ammonia, but on the right-hand side, if you look at the nitrate production, urea is going to produce much more, or substantially more nitrate than all the other co-crystal formulations of urea itself. So the histogram on the far right-hand side, you want that to be as low as possible, because that will indicate that we have limited the production of nitrate, which is something that we really, really want to try to accomplish. So even though the hydrolysis, which is shown on the left-hand side, is not necessarily limited by the co-crystals, the nitrification process, which is indicated on the right-hand side, is substantially limited if we administer urea in the form of a co-crystal of that substance. So a couple of take-home messages here. The hydrolysis shows initially a very, very slow inhibitory effect by the co-crystals. If we leave that in for a longer period of time, however, the co-crystals does exhibit some slowdown effect of that urea hydrolysis. But the more important part, and that's the inhibition of the nitrification process, that's the second step in this cycle. And essentially all the different co-crystals of urea substantially slow down the nitrification process, which means that the window of opportunity for the plant to absorb or take up nitrogen is substantially widened. And this potentially could lead us to far, far superior ways of managing that whole nitrogen cycle. So in conclusion, then, we have shown that the co-crystallization technology has helped us to maximize or optimize the way in which we can transform urea into nitrogen-containing compounds that are better suited for the plants, and that substantially minimize nitrification, which is one of the key steps that will ultimately produce a series of greenhouse gases and unwanted fertilization of freshwater sources, for example.
this has been shown by the detailed studies from the Department of Agronomy in their soil incubation studies. And the combination of the two departments and very, very different technologies that haven't been combined before now beginning to show real promise as a way of transforming these kind of very, very fundamental scientific tools into some practical opportunities for commercialization for new crop protection or production, not just nationally, but globally as well. So very, very last thing then. So there are some serious geopolitical realities that obviously are going to impact food production as well. And if you simply search for global food systems and fertilizers, I've highlighted 3 different quotes here, and all of them are really, really quite disturbing, because a spike in prices for fertilizers will lead to higher food costs, lower productivity, and there's a whole range of reasons for why we need to try to optimize the way in which we can manage and deliver nitrogen to plants. Finally, I'd like to acknowledge the work done by the Hettiarachchi group and Rwandi Kowarasinga, who is a postdoc in Ganga's group in agronomy. She did all the soil incubation studies, and Kelly Shunye, who's a graduate student in my group, did all the synthesis of the co-crystals. And I'm really, really happy about funding a seed grant from Global Food System, because otherwise we would never have started this collaboration at all in the first place. And at the very, very bottom there, you have some contact information if you need further information, either from the Department of Agronomy or from the Department of Chemistry. And if you have any questions about any of this work, feel free to contact either of us. And thank you very much for your attention.